So back to where we were cut off, we were talking about the idea that multiple RNA polymerases can be transcribing a gene at the same time. So hundreds of mRNA are created. So that means the RNA polymerase works a lot faster than DNA polymerase. And then another reason it works really fast is because all that proofreading that happens in DNA replication, that's not done in transcription. And the reason why is because it's not a problem. And I'll explain why it's not a problem. Think about replication. If those errors were not caught and replaced and fixed, then they would become permanent mutations. However, an error that occurs in transcription just means that that one individual mRNA molecule is going to be altered and going to be incorrect, which, yes, could create the improper protein. But there are hundreds or thousands of mRNA molecules. So who cares if just one out of the 100 are incorrect? The other 99% of the proteins are going to be fully functional. So it's not a problem. Not like it would be if it was DNA replication. So we'll move on. Now, once that mRNA molecule is made in transcription, it is not ready to move into the cytoplasm. There's a couple of reasons. If it went into the cytoplasm now, it might get chopped up by enzymes that are there. There are enzymes that are in the cell that are designed to break down nucleic acids. So if it goes into the cytoplasm, it could get chopped up. So there's got to be some modifications done to it to protect it from being chopped up. The other thing is when it moves into the cytoplasm, the mRNA right now, even though it's been made, there's parts of it that need to be removed. Otherwise, the protein is not going to be functional. And we'll see more about that coming up. So all of these things that happen to the mRNA after transcription but before being moved into the cytoplasm are called post-transcriptional modifications. And it should be noted that these only occur in eukaryotes. They do not occur in prokaryotes because prokaryotes don't have nuclei. There's not like a set spot where transcription and translation happens. It happens all in the same general area because prokaryotes don't have nuclei. So before these modifications occur, we could consider the mRNA to be a precursor or immature mRNA. And once it's done, it's a functional mRNA. It's a mature mRNA. So the first thing that's done is a 5' cap is added. So you can see the mRNA molecule here. There's the 5' end, and there's a cap added. This is the cap. And what is added is something called a modified guanine nucleoside triphosphate, or GTP, but it's modified because it has a 7-methylguanosine group added to it. And what the function of it is it protects the mRNA from digestion from nucleases and phosphatases that are in the cytoplasm. So I mentioned enzymes that break down nucleic acids. They're in the cytoplasm. Having this cap is going to protect it. It has an additional role of initiating translation. I do not expect you to memorize the structure of this thing. Just know that it's a 5' cap, so it's added to the 5' end. Know the name of it's added. So it's a GTP that's been modified with the 7-methyl group, but you would not be asked to draw the thing out. And definitely know what its function is, those two functions. So that's the first thing that happens. The second thing that happens is at the 3' end, at the other end, another modification happens. About 200 adenine ribonucleotides are added with the assistance of an enzyme called poly-A polymerase. So it adds this poly-A tail. It's called a poly-A tail. And whoops, if we go back, it is believed the function is not as clear, but it's believed the function of this is to maintain the stability of the molecule. To again, protect it from being broken down. Now, the other thing that's done is something called removing introns. So that molecule that was created with transcription, the gene actually has regions within it that are not meant to be turned into protein. They're called non-coding regions. So those regions are called introns. They need to be removed. And then the exons, which are the coding regions, which are meant to become protein, need to be put back together. So there's a process called splicing, which is going to cut out the introns and put together the exons. So I mentioned the small nuclear RNA. In splicing, it combines with proteins to become something called snRNP or SNRPs. They bind to an area between the introns and the exons, and they cut out the exons. And once it is associated with other proteins, becomes something called a spliceosome. It's going to cut out those introns and join the exons together. So it's probably easier just to use a diagram to explain this. So here is the mRNA molecule, right? Now, it's important to note that not all of this mRNA molecule that was created in the nucleus is meant to become protein. There are regions within it that are junk DNA, that are non-coding DNA that need to be removed. So this is the formation of the spliceosome, which is going to cut out this intron, so it's removed. And then the exons, those two pieces that are meant to turn into protein, are pieced together. And now you have a mature mRNA molecule. And this leads to something called alternative splicing. This is where the exons can be spliced together differently. So you have a gene. Here's the gene. The green is the introns, the non-coding material. The red is the exons, the coding material. And you can see that there's 4 different sections of coding material. So when there's splicing occurs and when the introns are cut out and then the exons are pieced together, yes, in a lot of cases, all 4 of the exons are pieced together to create what will become the protein. But in some cases, only, say, 3 of the 4 sections are put together to make one particular protein, and another 3 of the 4 are put together to create a different protein. So this is an example of alternative splicing, which is really an exception to the one gene, one polypeptide rule. So now you end up with an mRNA transcript that's ready to leave the nucleus and be turned into protein by the process of translation done by ribosomes. So again, it's important to emphasize there's no proofreading, which means there's more errors that happen in transcription compared to DNA replication. But you're doing so much transcription of those genes that it's not really a problem for the cell. So here you see the entire process, an overview of it. You see the 5' cap to poly A tail, the splicing that occurred. I'm not really going to lecture too much to this last section on HIV. The only reason I really have it in the PowerPoint is because HIV is considered a retrovirus. You would have learned about these last year. It's an RNA retrovirus, which means that its RNA must be converted to DNA so that it can hijack the cell, so it can become a part of the host genome. So it really is different from other viruses because it contains an enzyme called reverse transcriptase, which is able to convert the RNA to the DNA. And then the DNA can be incorporated into the infected cell's genome. So that's an important feature of retroviruses, the fact that they can do reverse transcription using this enzyme. And here you can see just an image showing the viral cycle of HIV.